Thank you. I'm so glad that all of you are here this evening, and we're very grateful for the Waikiki Aquarium folks for partnering with us. And I want to just give a special shout out to Mary Lou Foley, Nancy Taylor, and Dave White, who actually forged this relationship with Punahou School. So we're very grateful to have all of you here this evening for the Distinguished Lecture Series. Professional programs at Punahou is an opportunity for Punahou School to reach out into the community and also to take researchers, scholars, scientists, authors, and bridge the relationship between theory and practice. And so tonight, we're very lucky to have Richard Pyle here with us tonight because he is a consummate researcher, and he does the research that I think a lot of middle schoolers and high schoolers dream about, to have a job that you can put on that fancy equipment. Thank you, John Lee Bell and Ocean Hollis for bringing that to us this evening, to put that on your back and then go into the ocean and explore just an amazing world. And that's unusual for high schoolers and middle schoolers to see a real live scientist doing that real life work. A special shout out of thanks also to our communications office people who are here, our IT people who are here this evening, and Renee Chang, who actually did all the logistics to get everything in place for this evening. I have the distinct honor and pleasure of introducing Dr. Richard Pyle to Punahou School and the Distinguished Lecture Series here for the Waikiki Aquarium. And what you may or may not know is that Richard Pyle graduated from Punahou School in 1985, and he hasn't been back to the chapel in 30 years. So those of you who are in high school and junior school here at Punahou come every cycle, so he's having flashbacks. And the cans beneath the altar are not what he feeds his fish that he captures. It's the food drive. So if you are so inclined, please bring some food for the food drive this month for the chapel. Dr. Pyle is one of the first scientists, or among the first scientists in Hawaii that used rebreather technology, maybe even in the world. And my husband is also a diver, not to the extent that Richard is, and they actually dove together. And diving at these great depths is pretty challenging, and the deepest dive that Richard has done is over 500 feet deep, and that's a lot of water on top of you. So it's pretty remarkable. Only a handful of people actually know how to do it and can do it, and it takes great skill and talent and patience and confidence in your own ability to survive to do that sort of thing. So thank you for exploring the Twilight Zone for us so you can share it with us. And I'm not going to share any more details about Richard because he is going to share his journey as part of his talk this evening. Yet you should know that he truly is a scientist. He works at the Bishop Museum, and he also has collected over 20 and captured, I guess, over 200 species of fish that had previously been unidentified. And he also is a wonderful person. He's a husband. and he is also a consummate teacher, and he loves sharing his diving experiences. So thank you for coming this evening, and a warm welcome to Dr. Richard Pyle.
Wow. Thank you. Thank you very much. So thank you, Ruth, and thank you, Mary Lou, for organizing this, and thank you all for coming. As Ruth mentioned, I graduated from here in 1985, and I guess I was a reasonably good student, not terribly good student, but reasonably good. But I think my worst class was history, unfortunately. And I think the reason why I didn't do so well in history is because as a teenager, I didn't really have much history of my own, so I didn't really appreciate the importance of context that history gives us. And as an adult, I find history actually to be the absolute most fascinating thing on earth, even more than marine biology, you know, perish the thought. So if you'll indulge me, my talk tonight is actually framed around two primary stories, and those stories both relate to history on different time scales. And the first of those two stories is a very personal story, and it began almost 100 years ago exactly, when this guy here came to Hawaii for the first time. And his name was Edwin H. Bryan. Jr. And a couple years after coming to Hawaii, he started his 66-year career working at Bishop Museum. I believe he's worked longer at Bishop Museum than anyone in history. I don't know that for sure. But starting in 1919 is when he started working at Bishop Museum. And he's actually an extraordinary individual. He was an entomologist, he was a botanist, he was a cartographer, he was an astronomer. He just did all sorts of things, amazing things. He's probably best known for starting this series of books called Bryan's Sectional Maps. This is the first one, published in 1943. This is this year's edition, 72 continuous years of publication. But more than just that, he actually was an amazing scientist and an amazing explorer and an amazing naturalist. He covered the Pacific Ocean probably like no one before him. In fact, he's well known for coming up with the list of all the island names throughout the Pacific. He just traveled extensively throughout the Pacific. But I want to tell you something else about Ed Bryan that I don't know, I feel a little uncomfortable saying this, but in addition to being a great naturalist and explorer, he was also kind of a nerd. That's him on the right in that picture up there, and if you compare him to the dashing young couple on the left and the gruff captain of the ship in the middle, he's kind of geeky looking, and his pants are hiked up just a little too high. And if that's not enough proof, I'm going to quote from Neil Evenhuis's recounting of his 1924 expedition. After he passed away, when cleaning up his office, 2 small boxes were found in his desk. One was labeled, quote, "pencils too short to sharpen," and the other was labeled, "string too short to be tied." So Evenhuis went on to say, this is all he figured he needed to do to justify keeping it, was labeling it as useless. Now that's something that resonates very strongly with me, maybe more than I should admit. So he had a daughter. His daughter's name was Leilani, and his daughter definitely inherited his spirit of adventure and exploration. Not so much the nerd thing, though. This is his daughter, older in 1954, on the cover of Hawaiian Life magazine, covered in which I assume is mud. She was a surfer since the age of 8 years old. She was very outgoing. She preferred being outdoors, hiking, consummate hiker. She loved birds, she loved plants, she loved just exploring nature. And although she really wasn't much of a nerd herself, she definitely ended up marrying one. This is Bob. They met here in Hawaii, and they got married. With Leilani, Bob enjoyed the outdoors as well. They liked going hiking, and particularly Bob liked birds. And some of you may know Bob and know of his history with birds. And he actually left his mark here in Hawaii pretty amazingly through his bird endeavors. But they had 4 children, and the youngest of those 4 children, as you may be guessing by about now, is me. Turned out to be sort of a mix of all of those characteristics of nerddom and exploration. And so my very earliest childhood memories involve, stomping around tide pools, poking at things, lifting over rocks, being terrified by creatures that crawled out from underneath them. And through the years as I grew up, this is me at age about 13 in Micronesia, I got to know the Waikiki Aquarium. And I think of the Waikiki Aquarium as my other childhood home. I practically grew up there. And the lady on the right here is Ann Fielding. Some of you may remember her, know her. She used to run regular trips to Micronesia through the Waikiki Aquarium, and my parents and I went on several of them. And that's when my passion for ocean and coral reef and exploration really got solidified. In fact, I continued working at the aquarium, volunteering at the aquarium through my years here at Punahou, all through high school. Now, I graduated from Punahou, as Ruth said, in 1985, and that was the same year Ed Bryan passed away. I didn't know him very well at the time, even though he was my grandfather, but obviously I inherited some aspect of him. This is my high school graduation page. Now, it turned out I was in Christmas Island with my parents. They were on a birding tour, and I was dragged along when the seniors of my class did all the layout for their senior pages. So my 3 friends here created my little section of the yearbook for my graduating class. And I have to say that in 30 years of hindsight, they actually did a pretty amazing job. So first of all, they had a picture of my home aquarium, and they acknowledged my passion for fish that way. They had a picture of me in my scuba gear, and I don't know if you can see that, but I'm about to dive into very dark, unexplored waters in my parents' toilet in that photograph. But my favorite part is this fictitious quote that they invented as if it was written by me. After years of strenuous work and having surmounted excessive amounts of aquatic wildlife, I have traveled to yet another less complex part of the world to go where no man cares to go, to do what no respectable man wishes to do. I owe my thanks to my mother, my father, and my dog, Cisco. And I did have a dog named Cisco. Long live the Aquarium, Megamouth, and Jacques Cousteau. So I had nothing to do with writing that, and for some inexplicable reason, they came up with a pseudonym, Puce Dewey Oxnard Pyle. So this is, and yes, that is a great white shark tooth around my senior year neck picture there. So I graduated here, and I went on to the University of Hawaii as an undergraduate, still very much a nerd. And I worked for I went to one semester, and during that one semester, I had my very first job, which is a student aquarist at Waikiki Aquarium. And I was still trying to figure out what I wanted to be, and the same sort of thing that drove my grandfather out of California to come to Hawaii when he was 17 or 18 years old was driving me to do something different. So I dropped out of college, and I went off to go live in Palau. And one of the people I got to meet and got to know while I was in Palau was another nerd explorer. In fact, this is the uber fish nerd of all time. This is Dr. John E. Randall, Jack Randall, who's still at Bishop Museum. I took him for a dive on his 90th birthday last year. And Jack is really amazing. Jack has discovered and named and documented more species of coral reef fish than anyone in history. Not just than any other living ichthyologist, but anyone in history. And the reason he was so successful is his career began at the dawn of a brand new technology called scuba. So he was one of the first people to be able to take scuba underwater and explore fishes in their own realm, and that's how he was so successful. And he was a very big inspiration to me, and I wanted to be like him. I didn't want to be stuck in class and taking notes. I wanted to be out there doing adventurous, exciting things. And so I went to go live in Palau. And while I was living in Palau, at the age of 19, Jack Randall came down for a visit, and I took him out diving, and we went diving, and he took this picture of me. 2 days later, after this photograph was taken, I was quadriplegic. I had gone too deep, stayed too long, and come up too fast, and I had a very bad case of decompression sickness, the bends. It took me a year before I could walk normally again. It was a pretty harrowing experience, but it was probably the most significant pivot point in my life, heading me down the course that I'm on now, ironically enough. So that was a big event, and I realized from that experience that two things. One, there's lots of really cool stuff to explore down on these deep coral reefs. The reason I was going so deep that got me into trouble is Jack Randall had done all the exploration that one could do sanely with conventional scuba, and so in order to find something he hadn't found before, I had to keep going deeper and deeper. So I'd been bitten by the bug. That passion was in me. But I also knew that just using regular conventional scuba to do these dives was not the answer. So it was that incident that actually led me down a path to start exploring coral reefs. So first, the epiphany I had was basically scuba divers, even the most intrepid scuba divers, are limited down to about 50 meters depth, 165 feet with conventional scuba. Below that, breathing air creates all sorts of problems. You as divers, any of you who are divers will know this. If you're not divers, come up and ask me and I'll explain the physiology. But basically, going deeper than that, breathing air is very, very dangerous. So to go deeper, marine biologists use submersibles and submarines. And the problem with submersibles is there's a couple of problems with them. They're extremely expensive. And you can't get into a coral reef. You can't go into a cave with a submarine. You can do incredible things with them, but you can't really explore a coral reef very effectively. So there's this zone that's shallower than that's deeper than where scuba divers go. But since most submarines, if you're going to spend thousands of dollars to use a submarine that can go 2,000 feet, you're not going to mess around on a couple hundred feet. Most submarine stuff is done very, very deep. That leaves this zone in the middle here between about 50 meters and 150 meters, about 165 to 500 feet, that's very much unexplored. So we first started calling this area the Twilight Zone, which was like the perfect name for it, because up above you have these brightly lit shallow coral reefs. Down below you've got this perpetually dark abyss, and here's this twilight zone in between where it was literally a twilight zone. You go there in the middle of the day, it seems like it's about sunset time. That's how much light's down there. And also it was mysterious, and it was a great name. But the problem is it wasn't science-y enough, so we didn't really want to call it Twilight Zone anymore. So we started calling it Deep Coral Reefs. But It turns out around the same time they discovered these coral reefs that live thousands of feet deep, completely different kind of ecosystem, and those people started calling those reefs deep coral reefs. So we needed another term, and I didn't really have one, so for many years we started calling them deeper portions of typical shallow tropical coral reefs, which is a bit of a mouthful. And then thanks to the federal government, they had a workshop. They said, this is an important zone. We must explore it. We must give it a name. And the name shall be Mesophotic Coral Ecosystems, which is about as unexciting as, the opposite end of the spectrum from Twilight Zone. But that's what we're stuck with. We're now calling them these things Mesophotic Coral Ecosystems. So why not use a submarine to explore them? Well, we can, and in fact, we have. We had funding from NOAA for several years to do that off of Maui here, and we did a number of dives. Now, as I mentioned, submarines are extremely expensive, $30,000 to $40,000 a day to use one of them. But there's another limitation of submarines, which is that you are inside this big metal sphere looking through a little porthole trying to study tiny little fishes and tiny little invertebrates living in a very complex environment. And so while it's true that you can spend 8 hours in a submarine staring out these windows and taking videos and using a manipulator arm, you don't have the level of intimacy you do as a diver to try to explore the true inner workings of a coral reef. So knowing this as an undergraduate, I had to go back to the university for my dad's insurance to cover my bills for being paralyzed from the bends. So that's how I got back into academia again. And during that period, it's funny how life kind of twists and turns that way. I turned to high-tech scuba. Now, that does not look very high-tech. But what matters is not what it looks like on the outside. By the way, that rig was used for a dive to 300 feet. It wasn't by me, somebody else. But anyway, what's high-tech about this is not what's on the outside, it's what's on the inside. Those tanks contain different gas mixtures, not air. We're all breathing air now. But to go deep, you basically have to come up with different kinds of gas mixtures, usually involving helium and different amounts of oxygen. So using this mixed gas technology, I got more and more sophisticated. I designed this rig with 4 scuba tanks and 5 regulators, and we used it on an expedition to the Cook Islands in the 1980s, and down there at 300 feet, and this photograph actually was taken by that guy wearing that other rig of me down at 300 feet, catching all kinds of amazing new fish. Now, this is one of the first new species we discovered, and this is a very special fish for me. I named it. It's called Centripigi boylei, along with my advisor, Jack Randall. And the reason it has such an ugly name, boylei, no offense, Chip Boyle, it's named after Chip Boyle, who is the guy who first discovered it, who I was diving with there at the time. It's also kind of a cool fish because there's one of them alive right now down at the Waikiki Aquarium that I caught many years later in Moorea. So it's a really spectacular fish. It's just one of the many new species we found. This is another one. This is actually my favorite fish of all. This one's called Balanaperca Pyle, and it's not the reason why it's my favorite fish. It's actually just this incredibly charismatic fish. It's an amazing, it looked like, we didn't know what it was when we found it, so we didn't know what to call it. We just started calling it the Dr. Seuss fish because it looked like something out of a Dr. Seuss book. It turned out to be a soap fish. But anyway, this is another one. And this is another one that's near and dear to me because we actually first collected this one while breathing air at about 110 meters deep, which is sort of way beyond the limits. And if you're a diver, you know that there's a malady you get breathing nitrogen that deep called nitrogen narcosis. And it's like alcohol inebriation. And so because of our state of being when we collected the first specimens, when I first collected this, I actually forgot I had collected it by the time I was coming back up to the surface. And I thought, as the fog's lifting, as I'm starting to sober up after this dive, I thought I saw this yellow fish with a black spot, and wow, I wish I'd caught it. And then like an hour later, I looked in my bucket, I had caught it. I just forgot it. So this one, inspired by my good friend Randy Kosaki, who's here tonight, got named Centripigi narcosis in honor of the state of mind of the collector, which wasn't too different from what that picture looks like there. So I started turning to a new technology. I knew there had to be a better way, and this is the better way. It's called a closed-circuit rebreather, and there's one of them right outside on the table out there. It's actually old technology, but it's been revived in the last couple of decades because of modern electronics. They're smaller, they require less power, they have more reliable oxygen sensors. So I spent much of my graduate and undergraduate and graduate career thinking about designing these rebreathers, and over the years actually ended up helping this company, Cislunar Development Laboratories, go through. I started with generation 4 of their prototype. This picture up there is of the Mark V, and then there was a Mark VI, and now we're using something called the Mark VII, and we're already starting to design the next generation. Now, just to emphasize what a nerd I am, this is the Mark IV. This is my daughter, age 2 days. So what kind of guy brings his daughter home on the first day, straps her into a rebreather, and snaps a picture? And then this is her 19 years later. The rebreather got a lot smaller. She got a bit bigger. So anyway, over the years, I had this tremendous, part of my life is helping to design and devise these high-tech scuba gear to allow us to go down deeper, stay longer, and be safer on all of these dives. And it's been really, really exciting part of my life. It's more than just biology. It's engineering. It's really, really exciting. And a lot of what you're seeing up here are various prototypes that we tested over the years on our expeditions. So being connected to this company, I had access to this cutting-edge technology to allow me to use it for my biological research. And these are the places we've been over the last 20, 25 years or so throughout the Pacific. Now, I haven't caught up with Ed Bryan in terms of number of places he's been, but I'm slowly getting there. I've got a few more years to go yet. But these are the places we've taken this high-tech technology and gone down to explore the deep coral reefs. And this is an example of one of our recent expeditions a couple of years ago in the Philippines with the University of Hawaii. Sonia Rowley, who's here in the audience tonight, and a bunch of other friends of mine were down there. And over the years, this is just a small sampling of the new species we've discovered. These are the ones that I happen to have reasonably decent photographs of and that we're reasonably confident are indeed new species. As Ruth said, it's getting close to 200 new species of fishes we found down there. And what's truly extraordinary is if you do the math, how much time we actually spend at those depths, we're finding on average about 12 new species per hour of dive time. Now that's fish. Now, there are places in the Amazonian jungle where you can find insects probably at that rate, but there's nowhere on Earth you can find vertebrate new species at that rate. Before we started using this high-tech gear to explore these deep coral reefs, most biologists had assumed we'd found just about all the coral reef fishes there are, because most biologists didn't realize how extensive the coral reef habitat was below the depths where scuba lets you go. And when we opened up the door, now we realize we probably only have about 1/2 to 2/3 of all the known species of coral reef fish discovered. The rest are waiting on these deep coral reefs for us to go find and discover as we go. But it's not just new species we're finding either. One of the unexpected things we discovered traveling all across the Pacific is very different patterns of biogeography, that is, where species live across the Pacific. The rules are different down deep than they are shallow, and my colleagues and I are trying to figure out why. We just submitted a paper sort of outlining a hypothesis to explain it, but one of the key points is that down deep, you have higher rates of endemic species, endemism. So endemism means it's a species found only in one place. So if a species is endemic to Hawaii, it means it lives in Hawaii and nowhere else. So if you look at shallow coral reefs in Hawaii, about 25% of the species, plus or minus, on the shallow coral reefs are species that are only found in Hawaii. This photograph taken off Maui at about 350 feet, there are 2 fish in this photograph that are not endemic. Every other fish in this photograph is a species that's endemic to the Hawaiian Islands. And this photograph, which we just took, Randy Kosaki and I did a dive at Kure Atoll at 300 feet just this past September. Every single fish in this photograph is a species endemic to the Hawaiian Islands. This is far and away the highest rates of endemism in the marine environment ever documented anywhere. So keep in mind that before we started exploring this, the highest rate of endemism known for fishes in the coral reef world was Hawaii at 25%. Now we're tipping the scales at nearly 100%. So we've essentially quadrupled the highest rate of known endemism. So that's important. What that means is these deep coral reefs don't just harbor a rich diversity, they harbor a unique diversity, species found only in certain parts of the world. So that's exciting. And what's really, really exciting is that fishes are just the window dressing. They're just the tip of the iceberg, because the real biodiversity on these deep coral reefs are invertebrates. And this picture was taken by Sonia Rowley. It's an incredible riot of biodiversity. And Sonia's expertise is on Gorgonian corals. So what you see down there, you don't see a lot of fish when you take pictures like this. Sure, there are fish, and sure, we're finding new fish. But what truly dominates this environment are the invertebrates. So this is Sonia down at about 400 feet collecting a specimen of Gorgonian. These are examples of Gorgonians. This is another picture she took in Ponape. And what's really neat about Gorgonians, besides the fact that they're really cool story behind them with corals, but they're entire communities unto themselves. They're not just a coral. They're all kinds of organisms that live in association, direct association with the Gorgonians, including fish. It's a seahorse. It's a seahorse. It's tiny little pygmy seahorse. There's several new species of these things we found that mimic exactly the polyps. They're literally got these bumps all over their bodies that look just like the polyps of the Gorgonian coral. So there's layers upon layers, fleas upon fleas of incredible small diversity waiting to be found down there. So as you can imagine, as exciting as all this is, this kind of diving is actually quite serious. And so I'm going to show you, I have a short 4-minute video that kind of walks you through what one of these dives look like. And this actually is fairly accurate of what most of our dives looks like and how careful we have to be and how important it is to get all of this stuff right. So that's John Earl and myself. John Earl is another graduate of Punahou a few years before me, and his siblings actually taught here at Punahou, still do, I think, for many years. This is in Pyle. So in the upper right is what I'm saying, and the lower left is what John's saying. This was in 2007 filming for a BBC documentary called Pacific Abyss. We're 400 feet deep. So we're having this conversation. Very exciting new fish we're finding. So this video is cutting between John's camera and the BBC guy's camera. So you get the gist of what we're trying to do here. I'm trying to catch this butterfly fish called the Prognathodon, and then I'm going to get the angelfish after that. They're both incredibly rare fish. Here comes Bryan Green. Bryan Green's another guy who's been diving with us for years. He's younger than I am, but he's a really good diver. Bob is the BBC cameraman. See how seriously we take this. Here's Bryan. That's not the fish I was trying to catch. That was another fish that distracted me for just a moment. And then here's Bryan. Meanwhile, John Earl, the only serious one down there, has discovered yet another new species off to the side while we're grouping down. John's trying to get our attention to go catch this hawkbed. And meanwhile, we're playing around with this. So they're trying to catch the hawkfish. We never did catch the hawkfish. It's still a new species out there waiting for someone to find it. So the reason I show this video, not just to show how serious all this business is, but that fish that I caught 8 years ago is actually still alive in the Waikiki Aquarium today. In fact, that angelfish is is not only the only one in captivity in the world, it's the only one that's ever been in captivity in the world. And Bryan's ended up dying and mine stayed alive. So I think I eventually got the, sort of the, his is a great specimen in the museum. I'm happy to have it in a jar of alcohol, but mine is still alive and healthy 8 years later at the Waikiki Aquarium, only one ever collected. So that was kind of fun. That was a near and dear moment for me. Okay, so I'm going to change gears now. And I'm going to tell my second story. So I told you my first story, which is basically the context of where I come from. Now, the second story begins with me, or I guess I should say it ends with me. I'm going to tell this story backwards. And this is the ending of the story. This photograph was taken in 2011 off South Africa, 400 feet deep. And there are two large vertebrates who met each other down there. And the one on the left, as you probably guess, is me. And the one on the right is called a coelacanth. Now, some of you may have heard of a coelacanth. If you haven't heard of a coelacanth, look it up, because it's one of the most extraordinary organisms alive on planet Earth today. When it was discovered in 1936, it was the equivalent of discovering a Tyrannosaurus rex walking around somewhere in the Congo. And the reason for that is that fish had thought to have been extinct for 70 million years until a live one was found off South Africa in the 1930s. And there's an incredibly rich story behind it. It's not just some ancient fish. It's actually one of the earliest ancestors of all of us in this room. The reason I'm showing this to you is that, as I already explained, there were my parents. I was about 40 years old at the time. And it turns out coelacanths live to be about the same age as people do. And so let's just assume, for the sake of argument, this coelacanth I met was about the same age as me. And it had parents also, somewhere living, the same environment, 400 feet deep, somewhere off South Africa, about 40 years ago. So now we're going back in time 40 years to my birth and the birth of that coelacanth. So let's go another power of 10. So instead of going back 40 years, let's go back 400 years, all right? So it turns out my mom, who I forgot to mention is here tonight, Leilani is here tonight in the audience, is actually into genealogy. And she has, sort of researched our ancestry, so I can actually trace my own ancestry back generation after generation after generation, 400 years, and I end up with this guy who's Miles Standish, if you've heard of him. And that's, if you didn't flunk history in high school like I did, you would know that he was the leader of the first colony in the U.S. when the Mayflower came over. And the coelacanths don't keep genealogical records, but if they did, that coelacanth could likewise trace its ancestry back generation after generation after generation after generation, 400 years. So what you're looking at is sort of this history, sort of like how I started my talk, but on much larger time scales. Each node on there is a generation, okay? So now we're 400 years back. Let's go another power of 10. So now we're going to go 4,000 years back. And the same process could, in theory, happen. If I had records, if there were accurate records, I could trace my ancestry generation after generation after generation, 4,000 years back, and I would probably end up with some population of people living in Mesopotamia, and that's what Wikipedia says a guy living in Mesopotamia might have looked like. I don't know, I wasn't there. So my ancestors probably came from that part of the world around 4,000 years ago. And around 4,000 years ago, we have pretty good reason to believe that the coelacanths were living somewhere, in the Indian Ocean. We'll come to the coelacanth side of the story more. It's going to be boring for a little while here. I'm going to focus mostly on the left side. So let's go back another power of 10. So now we're going 40,000 years back. 40,000 years, generation after generation after generation after generation. If we had records, I could trace my ancestry back to people who were just beginning to colonize Europe some 40,000 years ago, and the coelacanth, if it had records, could do the same. So 40,000 years ago, there were coelacanths living off South Africa, just as 40,000 years ago, my ancestors were walking around in Northern Europe. Another power of 10. So now we're going 400,000 years ago. What was going on in my family tree 400,000 years ago? Well, we were, in fact, everyone in this room probably can trace their ancestry back to Northern Africa around that time. We were just really becoming civilized, the very dawn of civilization, the dawn of separating ourselves from other species. And in fact, that is on a time scale now that biologists now consider that to actually be a different species. So now we're talking time scales where my own ancestor was a different species from me. So that was considered to be Homo heidelbergensis. And it wasn't just my ancestor and everyone in this room's ancestor. It was the ancestor of actually the Neanderthals, our closest relatives that are no longer here alive, but they were. There was a time in history where our species and their species lived together, and both of our species could have traced our ancestry back. So and the coelacanth was doing something off there 400,000 years ago. Like I said, we'll come back to that side in a minute. All right, so now, going back another power of 10, so we're at 4 million years now. 4 million years ago, my ancestry goes back to this woman. You may know her as Lucy. And she's not only my ancestor and Neanderthal's ancestor and all of ancestors. She was actually the first human, or at least what we consider to be among the first humans, and she gave rise to lineages that split off, and now we're talking timescales where we got all kinds of species radiating. In fact, now she's even a different genus from us, so she's actually quite different 4 million years ago, and the coelacanth was a coelacanth. So I'll come back to that. I'm almost there. So another power of 10, 40 million years ago. Trace our ancestors back. We think they might have looked something sort of like that. Small little things living in the underbrush of the forest 40 million years ago. The dinosaurs were already gone at this point. They'd been gone for 30 million years already at that point. And it's not just our ancestors and humans' ancestors. It's the ancestors of all modern lemurs and monkeys and the great apes. And so I'm going to put this over on the other side, and you're going to wait a minute. How come that thing turned into all of us and all of those apes and all of those monkeys, but the coelacanth still looks exactly the same? How can I make that claim? How can I say that? Well, actually, there's pretty good reason for believing that. And the reason is this. There are actually 2 species of coelacanths alive today. In the 1990s, they discovered a second one in Indonesia. And that second one looked almost exactly like the one from South Africa. I mean, for a year, they weren't even sure if it was the same species or a different species because they looked identical. Well, recently they've done a whole genome analysis, and they found out that the estimate is these 2 nearly identical species of coelacanths separated about 30 million years ago. So if both living descendants of 30 million years of evolution still look exactly alike, you can be pretty confident that their common ancestor back in the day still looked alike. So it's kind of an interesting example of how diversification can be different and happen at different rates in different groups of organisms. All right, so now we're going to go back finally to the point where I meet the coelacanth before. So 400 million years gapped the time between when that coelacanth and I met in 2011 and the last time we were functionally together. And that was the common ancestor of both me and the coelacanth 400 million years ago. At least that's what the fossil record makes us think the ancestor looks a lot like. But we're not quite there yet. We're not at the beginning of the story. We still have one more power of 10 to go. Oh, by the way, that's not just my ancestor and the coelacanth's ancestor, it's the ancestor of every vertebrate. Remember I said before, coelacanths are on the track that led to vertebrates, not the track that led to fishes. So you'll see that all the tetrapods are up there, and they all derive from that. And just to give you a sense for scale and time, the entire age of the dinosaurs just fits in right there. So it's pretty big time we're talking about. But we're still, like I said, one more power of 10 to go. So now we're at nearly 4 billion years ago. We used to say life began 3.5 billion years ago, but they keep finding evidence that pushes us back further. So somewhere between 3.5 and 4 billion years ago, life began. And this is where the real crux of the point I want to make to you tonight is that every living thing on this planet today, everything from the bacteria to the fungi to the plants, everything has its common ancestor that goes back 4 billion years. We can trace our ancestry generation after generation. You know, you can see, we can all conceptualize that I am the child of my parents and my grandparents and my great-grandparents. We can get our heads around that. But that process literally continues back all the way to the beginning of life on Earth. So what all life is a literally unbroken chain of reproductive events going back that far. If you took one link out of those chains, we don't exist. And so it's really remarkable that this incredible diversity has spanned that time. And I'm just going to show you this one little set of graphics here to give you a sense for what kind of time we're talking about. So this guy, which was the second, wasn't the very beginning of life. This is the the second step, the second oldest in our ancestry tree here. So imagine you compress the history of life on Earth into 1 year, okay? The dawn of life was January 1st. This guy, way back then, when the coelacanths and I were the same species, Thanksgiving. That's where in that year this guy lived. If you took the history of life on Earth in 1 year, you'd have to wait till Thanksgiving before this guy came around, okay? Right about this time of year. This one, that rat, that rat-like thing, December 27th, 4 days before the end of the year. That's how long it took in the history of life on Earth to come up with that, okay? Another power of 10, Lucy. Lucy was around New Year's Eve, 4 in the afternoon, okay? The guy making fire, 11:08, 52 minutes before midnight, okay? Colonizing Europe, 5 minutes before midnight. Mesopotamia man, 30 seconds before midnight. Miles Standish, 3 seconds before midnight. And I, of course, came along at 0.3 of a second before we all started screaming and yelling it's a brand new year. So just to give you a sense, if you know, it's hard to comprehend 4 billion years, but we can comprehend a year. And that is how these powers of 10 scale out. And the reason why we have such diversity on life is because of that incredibly deep time. We don't really conceptualize time very well, but deep time very well. But that's sort of a way to explain it. So what makes biodiversity remarkable is that throughout all this history, there are somewhere on the order of 10 to 30 million species alive today. That's our best guess. We don't even know how many there are. That's our best rough approximate sort of estimate. And as I said, all of these species are interconnected through unbroken chains of reproductive events spanning incredible amounts of time. And this is where I sort of get to the big point of all of this. If you were an extraterrestrial, some of you may be, but if you were, and you came to this solar system, right? You know, and you found this solar system, which of these big planets circling this little star would you find interesting? Now, obviously, the one with the rings is kind of cool, and the really big one with the red spot. But actually, I think we'd all agree that any sort of external observer would realize that one of them stands out. And the first assumption is it stands out because it has liquid water, and everyone gets excited about liquid water. But water is really common in the universe, and all it takes to make it liquid is the right temperature. What truly makes Earth unusual, unique, certainly within our solar system, if not the whole corner of the galaxy, is the extent of biodiversity that inhabits this planet. Life, right? There might be life on some of the moons of these other planets. We haven't quite nailed the coffin shut on that possibility. But we do know that there's an extraordinary amount of life on this planet. And you just have to know that any sentient being would recognize the distinction between life and non-living chemistry as being important. So if we establish that life is important, we ask, well, why is it important? And why is it particularly important to us as humans and the future of humans on Earth, the future of humanity? Well, the way I like to explain it is to use the metaphor of a library. So imagine that every species, all those 10 to 30 million species, are actually, each one of them is a book. And each book, just as you find in a library, has a unique story to tell, a unique piece of information, something different. Now, there's obviously lots of books about quantum physics. So there's a whole bunch of books, and just in the same way that there's a lot of species of angelfish, right? They're all kind of closely related. They all have a slightly different take on quantum physics or on what being an angelfish means. But when you consider the entire spectrum of biodiversity, it's a massive library of information. And so we're not looking at just 10 to 30 million species. We're looking at 10 to 30 million stories. And what makes these stories so remarkable is that these are stories of survival. These are the stories that ended up enduring nearly 4 billion years of adversity. These are the survival stories. This is nonfiction stories. These is information that's incredibly valuable for all manner of things that we're only beginning to imagine. So if you think of biodiversity like a library, we humans right now, in our current state of technology and our understanding of the genome and our understanding of how all of this stuff works and our ability to read the genome and read and understand what that information, the power of that information, we're like kindergartners. We're a kindergartner group running through the Library of Congress. And what do kindergartners do in the context of the Library of Congress? They're utterly oblivious to the value of the information that surrounds them, right? I mean, there's Shakespeare, there's Homer, there's plans to build nuclear power power plants. And kindergartners don't care. They're pulling books off shelves and writing crayons in them and ripping pages out and making paper airplanes and doing all that kind of stuff. And that's kind of how we're kludging along with biodiversity right now. A few kindergartners, though, are starting to get to the see-spot run level of reading, and that's where we are as humans in terms of our ability to understand genomic information. And that genomic information holds incredible secrets, just like the Library of Congress that are as foreign to us in genomics as nuclear fission is to a kindergartner in the Library of Congress. But we're fast learners. We're incredibly fast learners as a species. Within 100 years or so, we will understand the meaning of this biodiversity. We will be able to reverse engineer chemistry that will cure probably every disease every human ever has had or will have. We will understand how certain plants can convert sunlight energy into chemical energy with 98% efficiency, unlike our current solar cells. So there is valuable information. And those examples I just gave are just the simplest, sort of basic concepts. The most valuable information in this biodiversity library is the stuff we cannot even comprehend yet. We will, though. We will comprehend it because we are understanding how genomics works. But there's a bit of a problem. And the problem is this. So let's take a global total biodiversity right now of roughly 20 million species. All right? We started building the card catalog to this biodiversity library in 1750s, okay? So 250 years ago. And here's the progress we've made in 250 years. That's the card catalog to the biodiversity library. That is how many species we have named. I told you we have, let's say we have 20 million total species out there. We've only documented 2 million of them. And so there's an incredible amount of diversity out there that we still don't know. It's like we've just taken the first couple of aisles of this incredible library and built a card catalog. In fact, by definition, we've taken the aisles closest to the entrance, right? By definition, the 2 million species we've cataloged are the 2 million easiest ones for us humans to discover, right? The ones that are still waiting out there are the ones that require fancy rebreather technology to get to, or other kinds of technology to access this diversity. And here's the real problem. The library is on fire right now. It's burning. And there's growing supporting evidence that we're at the dawn of what's known as the 6th great extinction. There have been 5 previous extinctions. The last ones took out the dinosaurs. The current one that's happening now, by some estimates, is much, much faster. Species are going away much, much faster. And what's scary about this to me is that for every species we know goes extinct, there's going to be at least 10 more that went extinct that we never even knew existed. We never even got the card catalog for. That's like burning, making a species go extinct is like burning the last copy of a book. And not only do you not have the information in that book anymore, but you never even knew that book existed. It's the Library of Alexandria times 1,000, times 1,000,000. So if we keep our current rate, we're not looking too good to be able to capture this information over the next couple of centuries in order to understand the true value of this information that surrounds us in this biodiversity library. So part of the world I live in is trying to figure out what happens there. How are we going to change this? How are we going to dramatically ramp up the rate at which we document biodiversity? And conservation is certainly part of it, and it's a very important part of it. Conservation is like putting out the fire, right? I mean, the library's burning, get some buckets and throw some water on it, put out the fire. And we're doing that. We're doing that real, real hard. The problem is if we lose that war and we spend all of our effort trying to put out the fire and we lose, then the diversity is gone. So what I do as a taxonomist, as someone who works in a museum, at Bishop Museum, is that we basically run into the library, grab a couple of copies of books, and run outside the library and put them in a vault. So the museum's like a vault. We're capturing these organisms, we're preserving them for future generations so that someday, maybe some future generation will at least have access to the genomic information that contained in those species, and maybe possibly Jurassic Park it back to life, who knows. But the point is the information won't have been lost, even if the species has been lost in nature. And so the big stumbling block right now is this. And so I'm going to do one last little series of sort of get your head around powers of 10. And it has to do with funding and funding the biodiversity research. So on this graph of starting off with a $5 million scale, the National Science Foundation had a program called Planetary Biodiversity Inventories, which was specifically to go out and do what I'm describing doing, document biodiversity, find, discover new species, get them into museums, understand that they exist before they're gone. And one of those grant proposals, the average grant proposal, was about $3 million. So that's a pretty big chunk of money. That's a much bigger grant than most biologists get, especially to do field research. All right, so let's squeeze this down by a power of 10. So now we're up to $50 million. And that right there shows you the total amount over the 10-year period that NSF funded these projects. They've sunk into biodiversity. So $30 million, that's a lot of money. National Science Foundation threw $30 million at trying to document biodiversity. That's awesome, kind of. Because if we scale down to another power of 10, this is the entire National Science Foundation Division of Environmental Biology budget, right, for a year. So that's putting things into scale. So it's a relatively small portion that goes into biodiversity research. This is a single space shuttle launch, all right, starting to put some things into context. So let's go another power of 10. So now our scale is $50 billion, okay? That's the entire National Science Foundation budget, $7 billion roughly, okay? That's the Large Hadron Collider. That's one project costs more than the entire annual budget of the National Science Foundation. It was an international cooperative project. That's why it costs so much. That's the Human Genome Project, $11 billion, right? Okay, so another huge, big science project. That's the annual budget of NASA. So now even NASA is sort of in the same league as this. That's the Manhattan Project in today's dollars. So that was the project that invented the nuclear bomb. And we got another power of 10. So that's the International Space Station. That's the space shuttle in total budget we've thrown for space exploration, and that's the Apollo 11 program. Now, I don't mean to disparage these kinds of research. I actually love space exploration like my grandfather. Astronomy is sort of a second calling in my life, and I think it's super exciting, and I want to see the money go for it. I don't want to reduce those green bars. But what concerns me is if you look at the left side of that graph, that's indistinguishable from zero. We're throwing effectively zero at this problem of the biodiversity library going, burning. And as I said, if you looked at the grand big picture, put yourself in the shoes of an extraterrestrial coming to visit our solar system, biodiversity is probably what would stand out the most. And what a shame that we're squandering it as kindergartners oblivious to its value. So my long-term preach, my soapbox speech, is that we have to really recalibrate our perception of the value of biodiversity, the perception, conservation is awesome. Let's try to save as much as we can. Let's try to put that fire out. Let's keep that fight going. But at the same time, let's at least get the card catalog built so we know what we had, and maybe a future generation will be able to at least access that information. Because if we never discover a species, if it goes extinct before we discover it, we never knew it existed. So this is a quote from my good friend Sylvia Earle. What we do or fail to do in the next 10 years will have a magnified impact in the next 10,000. And the idea behind that is that we are at a pivot point right now. We're kind of at an inflection point in history. We're kind of on the brink with climate change and extinction just sort of ramping up. We're sort of the first generation to get our heads around the scope of the problem, and very likely the last generation in a position to do anything about it. So what this means is that we really have to rethink our priorities in the long term, not just ourselves, not just the next presidential election cycle, not even just the next couple of generations, but I'm talking let's think big, let's think beyond our own lives, let's think about humanity over the next tens of thousands, hundreds of thousands of years. So I'm going to close tonight with Closing the Loop. This is Ed Bryan's great-grandson, Leilani's grandson, my son Owen, who's sitting right here. On his very first rebreather dive. I'm proud to say he's got nerd-like qualities, I'm sorry, and also exploration-like qualities. And so he mixes that same combination of genes that I think, and attributes that I think go on to greatness. So anyway, I just wanted to show that, and thank you all for your attention, and I appreciate it. Thanks.
Thank you, Richard. That was spectacular. I don't know if you know this, but I'm a secret paleontologist, and so that was just a magnificent display of change over time in our biology. So we share that in common. Richard will take some questions if you have any, or yes.
Thank you.
Love to know how you put that together and who helped you do it.
Many long nights, actually. I first put that together, that set of slides, those animated evolution slides, for a talk I gave in London a few years ago, and I don't think I slept for about 3 days. But thank you, and I actually completely agree with your point. I mean, I have some fellow graduate student friends of mine in this room, and we can all tell you that certain professors who are absolutely brilliant cannot communicate very well. And that's not a bad thing. I mean, there is probably a correlation between the quirks of personality that make one a genius in science that almost predisposes them to be inept at communicating those ideas. But the world does need translators, and the world does have translators. And there are a few people out there. There's some great, it's like Bryan Green, not my Bryan Green who stole my fish, but Bryan Green, the physicist, who's incredibly good at that sort of thing. And I totally agree with you. The reason people aren't passionate about these things is because they don't understand them, and the reason they don't understand them is the people who do understand them have a hard time communicating them. So I couldn't agree with you more on that. Any other? Yeah.
Could you talk a little bit about, I was really surprised to see you in just board shorts, like feet. Talk water temperature, and also, I understand it's easy to get down. Talk a little
Sure.
bit about ascent, which as a
No, great question. So first question on the thermal thing. I am a bit of a freak. And my friends in the room who've dived with me know this very well about me. I choose not to wear a wetsuit. Now, it used to be easy for me to do that because I used to have an 80-pound wetsuit that I used to wear on me 24/7 all the time. In the last few years, I've managed to lose that wetsuit, which means I now have to wear a little tiny thin wetsuit under my collared button-up shirt. But there's a bunch of reasons why I don't wear wetsuits. A lot of them is that it's easier for me to catch fish if my arms are unencumbered. I don't like the buoyancy changes that happen. If you spend $500 on a really nice wetsuit and do one dive to 500 feet, you're pretty much ready to throw it away because it's crushed. So there's a number of reasons that led me down that path, and my body just sort of adapted to not needing a wetsuit. But getting straight to your question, it can be quite cold down there. On our last trip to Punahou, I think we were getting down, Sonia, what was it, like the mid-50s or something, high 50s? Yeah. Yeah, quite cold. And it's something like 85 Fahrenheit, close to 30 centigrade up in the shallows. So it's quite a bit of a temperature shock. And there was one dive, a particularly memorable dive I made where it went from 85 degrees Fahrenheit to 55 degrees Fahrenheit in 1 meter. And that got my attention. So I guess I don't know if I really answered your question. Most people who do this wear dry suits, which are at the upper end of thermal protection. Most of my friends here in the tropics wear wetsuits, usually 1 or 2 or sometimes 3 layers. And then you end up paying the price in the shallow water because it can get a little hot, which actually comes to the second part of your question, the ascent part. So it takes us about 3 to 5 minutes to get down there. So we can zip down there pretty briskly. Coming back up is the tricky bit. And basically what it boils down to is when you're down breathing gas under that pressure, the gas that's inside your lungs that you inhale dissolves inside your blood and body. It's something called Henry's Law, which says that the gas dissolves into a fluid, in proportion to the space over it, which in our case is our lungs. And so over time, that gas accumulates in our blood in the same way that carbon dioxide accumulates in a soda under pressure. And in the same way as a soda pop that you take the top off quickly, especially if you've shaken it, froths with bubbles, that will happen inside of our body if we came up too fast. In fact, that's exactly what happened to me when I got paralyzed. I had what's called the bends, which basically boils down to you go down too deep, you stay too long, gas absorbs inside your body, and then you come up too fast without allowing time for this gas to come out of your body through your exhaled breaths. And if you don't allow that time, the gas has to go somewhere because you're removing the pressure that was holding it in, and so you've got all this excess gas in your blood and tissues that it has to go somewhere, so it forms bubbles inside your body, and those bubbles wreak all kinds of havoc, including paralysis and death. So to avoid that, we have these computer algorithms that track how long we're down, how deep we are, what gas we're mixing, breathing, and it then sort of monitors how much it guesstimates is inside our body and gives us recommendations for the rate of ascent. So a typical dive for us to say 100 meters, 330 feet. If we stay down there 20 minutes, we'd decompress for maybe 2 hours. If we stayed down there for an hour, we'd probably decompress 4 or 5 hours. The longest dives we do is around 8 hours, not because we're limited by the equipment. These rebreathers will actually let us stay underwater upwards of 12 hours if we want to, but there are other reasons why you don't want to be underwater for 12 hours. One of them is eating, another one's drinking, and there's some other factors that have to happen in a 12-hour span of time. So we generally try to limit our time. on the bottom, and the deeper we go, the more severe that ratio is. So if we're going down to 500 feet, we only have 5 or 10 minutes, and we're still looking at 3 or 4 hours of decompression. So we have to play that game delicately. We don't want to overcommit to being in the water for too long. It's just that the deeper we go, the longer we stay, the more time it takes to allow all that excess gas in our body to come out. And that ultimately is why I can claim such high rates of new species per hour, not just because we're discovering so many new species, but because we have spent so few hours down there. Now, there are ways to do it better. There's something called saturation diving, and the way saturation diving works is you leave your body at that pressure for weeks or even months. And this is what commercial divers do and military divers do. And you do have to decompress from that, and the decompression does take a couple of weeks, but the ratio of decompression time to bottom time is much more favorable. Instead of a few minutes of bottom time to hours of decompression, you could have 3 weeks of bottom time to 1 week of decompression. So you get a much improved ratio. But obviously you can't be underwater for weeks at a time or even more than a few hours at a time. So that kind of diving protocol requires chambers and submarines and pressure pots, and you basically sleep inside a giant pressurized chamber, huge ship, lots of crew, lots of money, like I talked about at the end. We biologists don't have anywhere near that kind of money to do that. We could, if we had that money, spend 8 hours a day, 7 days a week for a whole month exploring this, and we would do in 10 of my lifetimes in one project, but it requires a lot more funding than we have. So I think that addressed both of your questions. It took a while to get there, but yeah. Any other questions? Yeah.
What about fish that you're capturing down there? If you're coming up and compressing on the way up, they decompress?
So that's a good question. So the question is, how do the fish decompress, and do they decompress along with us? And to some extent they do. But actually, the good news is that fish have a different situation than we do. The reason we have to decompress the way I just explained we have to decompress is because we're breathing gas, and that gas is under pressure. That's where this Henry's law comes into effect, when you have compressed gas and a fluid next to each other. Fish don't breathe air, they breathe water. And it turns out the gas that's dissolved in the water they're breathing is at the pressure it was when it dissolved in, which is at sea level pressure, which means that the dissolved gas in water is just 1 atmosphere. It's not high pressure the way it is inside our lungs when we're breathing it heavy. So we have to decompress for a completely different set of reasons than fish. Now, having said that, most fish you cannot just bring straight to the surface for a completely different reason, which is that most fish have a little gas sac inside of their body called a swim bladder. It's a little sac of gas that allows the fish to not sink, so a sleeping fish can hover in mid-water. Because fish, when you add up their bones and their muscles and everything, are denser than water, so they would either have to swim perpetually to stay up, or they have a little gas bubble inside them that keeps them neutrally buoyant. But that gas bubble will expand as we ascend. So our solution to that is we take a hypodermic needle, same thing the doctor gives you a shot with, insert it into the side of the fish, we know where the swim bladder is, and the bubbles just sort of vent out of the swim bladder, and that way the fish can come right up to the surface. So we can actually get to the fish to the surface in minutes, even though we have to stay hours waiting for the gas to come out of our bodies molecule by molecule. Yeah. Hey.
I have to agree with the woman's comment earlier about just doing a tremendous job of communicating.
Thank you.
All of the amazing things that you've done in your career and your passion for what you've done as part of your career. It's just incredible. I'm trying to compress a couple of quick questions in one.
Okay.
Maybe I can capture this. Is you talked about the degree of endemism that you're finding in these depths, these Mesophotic Coral Ecosystems, and I'm along the lines of wondering, seeing your map of all the flags of the locations that you went, I'm sure there's locations you'd love to go. Is there a place where you may have a hunch or a clue where there might be not only high degree of endemism, because you're finding that already right here, but maybe even high proportion of diversity of
Endemics. That's a really excellent question. So we don't know yet. And the reason we don't know yet is even though there's stars all over the map, those stars collectively represent only a couple of hours of bottom time at each place, just because, if we're there for 3 weeks, if you add up the actual minutes we get of exploration time. So we're kind of only at the beginning of understanding where things are, and we're finding new things on every dive. So until we get to that sort of asymptote, we won't really know what the true big picture is of where the diversity hotspots are, where the endemism hotspots are. Hawaii is a bit of an exception because we've had much longer history of exploration happening here. We have had, the University of Hawaii has had a submarine operation here for many years. So we've had access to technology that's helped us explore Hawaii much better. Coincidentally, Hawaii also seems to be probably the endemism capital of the Pacific by a long shot. Easter Island might come close, but it's got low diversity. So I think the question you were asking is where in the Pacific might there be higher diversity in Hawaii and as high or nearly as high endemism? And I don't know. I really don't know. I mean, every place we go, it's fascinating. The other pattern we find, if you know coral reef biogeography at all, you know that the highest diversity places in the world are what's called the Coral Triangle. It's between the Philippines, New Guinea, and Indonesia. And if you look at a shallow coral reef there, there's maybe 2,500 species of fish that live in that region. And there's only 500 or 600 that live in Hawaii. And there's a very clean pattern, and this applies to corals and fish and everything else, clean pattern of less and less diversity as you get farther and farther away from that hotspot. Well, what we're finding is that pattern only applies to the top 100 feet or so of coral reef. When you get down deep, we find just as many new species of fish and unique species of fish way out in the Cook Islands or the Society Islands than we do in Indonesia or New Guinea or the Philippines. And so it's a completely different pattern. It factors into this hypothesis. So I guess what I'm getting at is we're still at the very early stages of just trying to get our heads around what's going on these deep reefs. There will be an answer to that question. It's too early for me to predict where it's going to be. Wake is like #1 on our hit list of places to go. The problem with Wake is it's really hard to get there. And there's military clearances you need and a few others. There's a few spots in the Pacific we desperately want to get to. One of them is that place where the Chinese are now building islands. And that is actually one of the most spectacular places we want to go because it has a combination of very clear water, which is useful, for these rich, deep coral reefs, and being way out there in the triangle, the Coral Triangle. But obviously, that's not a safe place for us to go show up with our little, rebreathers and our little dive boat and don't shoot us. We're just looking at fish. So there's some of the places we want to go but haven't been yet are because of some logistical reason, and Wake is definitely one of those. Another one's called Minami Torishima, which used to be called Marcus Island, and it's sort of out in that same direction as Wake. There's some weird stuff going on out there, and we'd love to go deep. And then another place is the Phoenix Islands, which is kind of dead center in the middle of the Pacific. That's another area we haven't been to yet, but we really want to get to. Yeah.
So what did that feel like when the seal came?
I cannot describe that feeling. I actually have video of it all because it was all being filmed. But I'll tell you a quick funny story. It wasn't funny at the time, but it's funny in retrospect. So basically, National Geographic hired us to go do this. And we had a guide named Peter Tim who had found the coelacanths off there, knew where they were, and he was taking us where to go. And we were diving there every day. 13 dives in a row, never found a coelacanth. Same patch of ledge. We'd always start here. We'd always swim along this same 200-yard stretch, look in every cave where they've seen coelacanths before. Never found it, never found it, never found it. The producers were getting kind of high-strung about it. We got to have footage of you and the coelacanth. And so we were getting towards the end of the trip, and I think it was the second to last dive. We did the same dive. We got to the same end of the same ledge and the same caves, and nobody home all the way along. And we were all on rebreathers, and this guy, Peter's old school, he was using mixed gas scuba. So he had like 5 or 6 scuba tanks on him, so he had all this massive stuff on him. But the problem with scuba is you don't have margin for error. Now, I told you a rebreather can give us 12 hours, right? Scuba, you're very limited in how much time you can breathe. If we have a problem on a rebreather and we need an extra hour of time, no big deal. Scuba divers don't get that margin for error. So he was on scuba, mixed gas scuba, and we were at the end of the dive, and the tricky thing about this place is we had to leave the bottom by 13 minutes of bottom time. If we stayed longer than 13 minutes, then our total decompression time would be long enough that the current would take us out into really rough waters. We had to limit our total decompression time to 3.5 hours. So we had this sort of 3D chess game we were playing. So we get to the end of the bottom time, we start heading up, and Peter's nowhere to be found. And I had last seen his bubbles coming up from behind me. We're waiting for him, I turn around, no more bubbles. His bubbles are gone. And I thought, oh my God, body recovery time, right? So, because he didn't have any margin for error, scuba diver can't capture their bubbles. If the bubbles aren't going up, what happened? So I start swimming out over the drop-off to go look for him, expecting to see an unconscious diver on the bottom. And instead I see a flash, and I thought, did his computer explode? What was that? And so I come over the edge, and I realize he's taking pictures of something. And I'm like, Peter, we got to go. What are you taking pictures of? And when the flash went off, there was the silhouette, the unmistakable silhouette of a coelacanth. And the funny thing was, National Geographic had asked me, I had a video camera, and the video camera picked up everything I was saying in helium voice, chipmunk speak. And they had asked me to sort of narrate the dives and describe my feelings and, all that so they could put it in the documentary. And so when I realized, I had rehearsed like what I would say, oh, behold, this great, evolutionary ant, I had some thing worked out. And if you hear the video, it's R-rated, but it's actually quite funny what I actually say when I first realize I'm looking at a coelacanth. I won't quote it, but anyway, it was actually a funny and exciting and exhilarating experience all at once when I saw it for the first time. That was the end of our dive, so we only had like 2 minutes. It was like get video, and we had the good fortune of seeing him on another dive later on. We spent a little more time with them, and at the very end of the last dive, we'd gone over our limit. We were already in trouble of getting into the rough water on decompression, and all the other divers are going up, and on my video camera, you can hear them going, come on, Richard, it's time to go, we gotta go. And I'm like, yeah, I'm coming. And it was just me and the coelacanth. And it was like this moment that I didn't have before because I was posing for the camera before, looking all sciencey. And then I had this one moment where the film crew had left already, and it was just me and the coelacanth. And he came past me really close. One thing I should explain is that my mental image of a coelacanth was of something maybe about that long and about this big around, right? That's what you see in books, that's what you see in pictures. This fish was 7 feet long and this big around. It was 2 orders of magnitude, it was 2 standard deviations out, not orders of magnitude, standard deviations out from what I had expected to see. These things are enormous. So his tail is the paddle about this big. And he came and he swam right past me. They're very docile, they weren't scared of us at all. And you can see on my video camera, my hand reaches out and sort of shakes, and I stroked its tail as it went by. And that was the moment that I'll never forget. That was unmistakable. I think we're out of time, but thank you again for coming. I really appreciate it, and thanks.
Thank you, everyone, for coming, and thank you, Richard. You're tremendous. Good night.